Okay, hello everybody and welcome. We're sorry we can't see you in person here at UNMC. Certainly we were hoping to have that opportunity, but obviously we have some difficult conditions right now. We hope that you and your families and communities are well. We are doing this live, so there could be some hiccups along the way and maybe we'll say silly things or make a mistake, but I hope you'll forgive us for that. My name is Scot Ouellette. I'm an associate professor in the Pathology and Microbiology department. And I'm here to answer some of your questions along with those of my colleagues. So I'll pass it over to Dr. Rucks.
Hi, I'm Lisa Rucks. I'm an associate professor in the Pathology and Microbiology department.
Hi, I'm Leah Cook. I'm an assistant professor in the Department of Pathology and Microbiology and also a member of the Fred and Pamela Buffett Cancer Center.
Well, thank you very much, the three of you, for joining us this morning. So we're just going to go through a few questions. This program is for Native American students that are going to tribal colleges and universities all over the country, and they'd like to learn a little bit more about research in general and from researchers about your careers. So let's go ahead and get started sharing some answers to the questions that they've asked. First is, let's discuss how long it takes to become a researcher. And if you can start describing your career path, would one of you like to get started on that?
Go ahead, Lisa.
All right, well, I found this to be an interesting question. So just to give you all some background on my education, I went to the College of Charleston as an undergrad. That's a small liberal arts school, so it's not going to be a big research institution. And so some of my initial research experiences, probably the best one, honestly, was in a plant physiology class. We had this great lab and we had to do experiments and we were in the greenhouse and that was great. And then for undergraduate research experience, I actually just emailed a bunch of folks who were right up the street at the Medical University of South Carolina, which was a biomedical research medical school institution. And that's how I got into my first research experience. So I think if I could just already plug a piece of advice is don't be afraid to cold email anybody if you're interested in their research. And the lab that I started doing my undergraduate research in ended up being the lab that I started my graduate school in. So I started as a student at the Medical University of South Carolina. We moved during my education. So my PhD is actually from West Virginia University, and that was a whole another layer of education, how to move a lab. I then went out to the Rocky Mountain Laboratories, which is in Hamilton, Montana, in the Bitterroot Valley, for my postdoc. And then my first faculty position was at the University of South Dakota. But I would say becoming a researcher really started in the undergraduate years and just seeking out opportunities there.
And maybe I'll jump in briefly then to say how long it takes depends on what level you want to be at. So for instance, we're all professors, so we had to do an undergraduate degree, of course. Then we did a PhD, and that's usually about 5 years on average, maybe 6, to do a PhD. And then oftentimes in our fields, there's a postdoctoral training period, and that might be another 3 to 5 years. And then you hopefully apply and can find a job as an assistant professor. So you can see it does take quite some time to get to that level of being an assistant professor in a research environment. But if you wanted to do an undergraduate degree, you could then very easily go work in industry, for instance, or in a hospital setting as doing research. It's just the amount of freedom you might have would be very different, I suppose. So it depends on what your personal goals are and what you're passionate about. And I think for each of us, we all probably had a lot of ideas and we wanted the freedom to pursue them. And that's why we went all this way to get to a professor level, because as a professor, we are free to explore whatever our interests are. Leah, do you want to talk about how you became a researcher?
Yeah, so I had a little bit of a non-traditional track record. I actually was attending a community college back home in Birmingham, Alabama. I was taking a break from a couple of years that I did at the University of Alabama, and I moved back home and started taking some classes to try to figure out what I wanted to do. And in that process, my biology teacher actually told me about this summer research program at University of Alabama, Birmingham. And so that was my first introduction to a research experience. And so that involved bringing in community college students into different labs at UAB. And so from there, that's where I really just became really interested in research. I had no idea about the entire scientific community, but I was put into a prostate cancer research lab where we were looking at dietary elements that can help prevent spontaneous prostate cancer. And so from there, I decided that I wanted to pursue a full 4-year bachelor's degree in biology, and I transferred to UAB and started majoring in biology. And then I knew I loved research, but I didn't know how far I wanted to go. And so I enrolled in what we had was a 5th-year master's program, which allows you to start working on a master's degree and doing research while you're completing your bachelor's degree. And there's a lot of those programs at different institutions, so that if you want to get an extended degree and still do research, you can do that. And so I finished that and finished my master's. And then in that process, kind of what you said, Scott, I then decided that I wanted to go further because I had my own ideas. I had my own research that I thought I really wanted to pursue, and I needed a PhD to do that. And so because UAB is a really strong place for biomedical research, I stayed and enrolled in their interdisciplinary biomedical research program. And from there, you pick a lab in your first year. I did my rotations, and then I joined the lab focusing on cancer metastasis. And that was kind of, the rest was history. I kind of was grabbed by cancer metastasis research and decided to then go on to pursue becoming an assistant professor. And so after I graduated, I went down to Moffitt Cancer Center in Tampa, Florida, and did a postdoctoral training fellowship for about 5 years. And then when I started looking for positions, I found out about this position at UNMC and applied. And so all in all, it probably was about 9.5 years. I tend to tell students, though, to be honest, anything that you want to do in the health professions takes extra training after you've gotten your bachelor's degree. And so it all ends up being about the same amount of time, regardless of which path you decide to take.
And it's not a race. I mean.
Right. Yeah.
Exactly. So can we start discussing, let's talk about how you got interested in your research and career path. I heard a little bit about what some of you, like Dr. Cook, what you were how you got interested, but how did you get interested in your particular career path?
To be honest, it was hilarious because we were asked what kind of lab we wanted to go to when we were applying for this summer program. And I remember specifically writing that I wanted to be associated with something related to food and how some type of food science research. And so then they put me into this lab that was a cancer lab looking at dietary extracts that can help prevent tumor growth, and also environmental factors that contribute to tumor development. And so that's actually how I got into cancer research. But I think what grabbed me was that, number one, every day was different. In my first week, I learned how to isolate proteins from tissue, and I'd never done anything like that before. And so it grabbed me because I was always a curious person. And so I was learning these new techniques, but also learning how tumor tissue comes together and different proteins that they make to be able to survive. And then also, I didn't know much about cancer. And so learning about how I could contribute to helping cancer patients survive was a big thing for me. It was a little bit of being involved in something that was bigger than me. And then also the third thing was that there are significant racial disparities in cancer risks. And so I saw it as an opportunity to be learning something, but then I could also take that information and help educate my family members. Prostate cancer, African-American men have the highest risk of getting prostate cancer. And so I immediately saw it as something that I could utilize to educate my dad and my brother and other Black men in my family, and also contribute to a bigger cause. And so that's what really kind of drove me to stay in cancer research.
How about you, Dr. Rucks?
Well, I think my interest when I started college, I was going to be a marine biologist. That was my dream. And then sophomore year, I took a comparative invertebrate embryology class, and that crushed all of those dreams pretty soundly. But at the same time, I was taking cell biology classes and molecular biology classes, and I was just fascinated. And so my first undergraduate research experience is we were studying bacterial toxins. And so we were taking cultured bacteria and mixing them with eukaryotic cells. And so you could see in real time when these bacteria would inject their toxins, these cells would round up. You could see this morphological change. And of course, as an undergrad, I didn't necessarily understand what that was other than it was really cool. And I was hooked on that. And so I was always fascinated by cellular microbiology. And so now I'm a chlamydia biologist. And for those of you who don't know, it's an obligate intracellular pathogen, which means that it grows inside eukaryotic cells. And so I wanted to become a better cellular microbiologist. So I thought, what better way to do that than to study an organism that you can't culture outside of anything but a eukaryotic cell? And so that was, the light bulb started turning on in some of my undergraduate classes. So I don't want to dismiss that, but then also reinforced by many of my experiences. Again, I'm going to cite the plant physiology class. Yes, we were in the greenhouse. We were also running SDS page gels and some agarose gels. So we were doing real research with real questions. And so that's how I was just constantly being curious and super excited about what was happening in the lab, especially because many of my early experiments I thought didn't work. I got a different phenotype in my experiments than any other person in the lab, but I was working with a different cell type. And so it was the first time that I ran a different cell type, a cell type that had been really well studied alongside the cell type that I was looking at. And I realized my cells were different. It wasn't me screwing up the experiment. My cells were different. And that was it. I was hooked. I knew I couldn't do anything else but research forever.
How about you, Dr. Ouellette?
Yeah, so when I was a high school student, biology was always my favorite subject. And so I knew when I went to college, I wanted to do something in biology. And I remember having a course about immunology that was quite interesting to me because I liked the aspect of the microbes coming in and then the immune system fighting them. And then, there's this battle being waged. And so that was kind of cool. However, I decided that I was more interested in the micro side. As far as how I got into research, that was also one of these, it was just chance in a way. So I think this is maybe where some of you are as well in terms of trying to think about how do you get into research, and Dr. Rucks alluded to it earlier. So I wanted to apply for a scholarship when I was in college, and it required research experience, and I didn't have that. So I went to my undergraduate advisor and said, well, how can I get research experience? Because I didn't really know how to do that. I'm the first scientist in my family, so I couldn't go ask any of my relatives about how do you do this? And I had nobody to talk to. So I went to my advisor and he said, well, this is the early days of the internet, but it was still there. So he said, go on the internet and look at some of the research that the faculty are doing in the department and see if there's something there that interests you. And then you should reach out to that person and ask them if they have an opening and the possibility of sponsoring you. So I started to do that. And as it turns out that my advisor actually apparently decided they needed somebody to come work in the lab, doing all of the, let's call it grunt work, washing dishes, filling pipette tip boxes, cleaning up in the lab. So, I jumped at it because I really wanted to have an opportunity. So that was my first experience in a lab. And it turns out that lab also worked on chlamydia. So Dr. Rucks works on chlamydia. She's just told you a little bit about that. And I have worked throughout my career on chlamydia because it's a very interesting organism that has a lot of unique aspects to it. So I basically worked my way up. So I was an undergrad washing dishes. That was my first job in a lab. And then I was paired with a graduate student and I helped her with her project. And then I eventually had my own project during a summer fellowship. I think that was between my sophomore and junior years. And then from that point on, I started to do my own projects in the lab. I did take a year off, a gap year between undergraduate and graduate school. And so I didn't actually know what I wanted to do. I mean, I liked doing research and I liked learning. So I thought, well, I may as well do a PhD because I get to do both of those things. And so I ultimately went to do a PhD. And then it was during my PhD that I thought, well, you know, I think I can do this and I have lots of my own ideas. So I think I want to have my own lab. And so that kind of set me on the path, which might answer a different question here a little bit later on. So I think you'd find when you talk to any researcher that we all have slightly different stories, but also slightly similar stories, right? So in many cases we may be the first scientist in our family, and we had to just reach out and talk to somebody and say, well, you know, I'd like to do research. How do I do it? You know, can I work with you? Do you have an opening? And it may not be at all what you thought you were interested in. So, you know, we have a lot of graduate students when they start their PhDs and they think they want to work on one thing. And then it turns out maybe they couldn't go into that lab. And so now they're working in a different lab and they become very passionate about that. So, you know, if you had told me when I was a young man or a young lad that I would be working on chlamydia my whole life, I probably would have said, you're crazy. I mean, why would I work on chlamydia? But, you know, that's how life turns out. So I've been working on chlamydia for over 20 years and I obviously really like it and I'm passionate about it and I think it's fascinating.
Can I make a comment to add to what Scott just said? One thing that really jumped out to me is that with also Lisa about your early experiences, and I think a lot of students think science and they think of what they see on the movies, and that if they go into these labs, they're going to be working on these groundbreaking cures for people who have a disease. And so when I actually transferred to UAB, it was because the PI that I was, or the faculty member that I was working for in the research program offered me a job as a work study student, but that meant feeding the mice and rats, cleaning up after them in the cages. And to be honest, it was a really gross job. And sometimes I had to work with these animals and I was really scared of them at the beginning, but I wanted to be in the lab and I just saw it as an opportunity to learn as much as I could. So I didn't even care that I wasn't on a project. And I didn't have a project for a while until I was encouraged to kind of go after the master's. But definitely, I think if you want to be in a lab and you want to be in research, be prepared to take some of the grunt work, but know that you're still going to get an opportunity to learn and just kind of see some of the research that's going on around you.
Yeah, definitely my first task in that lab, and she couldn't pay me money. So I was doing school and I had a job to help pay for everything. And in the time left over, I was in the lab. But I was stuffing pipette tip boxes. That was before we had the racks that you could just put in and make it easier. I was autoclaving. I was making reagents for the lab. And really the only reason why I was given a project is because there was a grant deadline coming up and she needed extra hands. And I had proven I wasn't a total disaster in the lab to begin with. So that was it. But yeah, we all kind of started at the bottom there.
And I think, you know, as a professor, when we have students who show an interest, that's a real positive thing. And we take that seriously. We want to give, you know, I think we all like to give younger people an opportunity in the lab. And so if a student comes to me and says, I really am interested in getting a research experience and I want to, you know, work with you, then I almost always give them a chance. I don't think there's a single time where I haven't given a student an opportunity. So that's something to keep in mind is when you're, you know, if you find something that you're interested in and you're hesitant to reach out to a professor, well, I would encourage you not to be hesitant because most of us, I think, would share the opinion I just stated. And not that there aren't those rare cases who might be kind of jerks and would ignore you, but you probably wouldn't want to work in their lab anyway. So look for a different opportunity, but because there are plenty.
Can you talk a little bit more about what type of personality it takes to go into research? I know that that type of job is really not for everyone, but can you speak a little bit more about that?
Well, I'll jump in because I think if you've listened to us, you'll probably pick up on the fact that we've all mentioned curiosity, right? So if you want to be a researcher and have some independence, you really have to have that curiosity, and you have to be driven and willing to work hard, of course. It's also helpful if you're very detail oriented, because when we do experiments, the goal of an experiment is to test one variable ideally. And so you need to be able to control all of the other things around you, which means you have to have that eye for detail. And so if you have that sort of obsessive compulsive disorder, if you have some level of that, that's an okay thing. It's an okay trait for a scientist. But again, depends on where your career goals lie, because if you want to work in industry or in a clinical setting doing research, then maybe those technical skills will be more important than the curiosity part, right? And maybe if you're interested in just doing that and being able to contribute and help, that's great. And so those types of skills will help for that. If you want to have the independence though, then you really have to maybe be curious and want to know why. And that's what drives, I think, most of us here is we see there's an observation, this occurs. Well, why does it occur? That's what we want to figure out. We want to know why. So you have to have that curiosity about how things work and want to get to the bottom of that.
Any other additions to that?
I think there needs to be some level of tenacity, because you will face adversity throughout your career. You're probably going to get more no's than yeses, whether that's in your experiments or the grants that you write or the scholarships you apply for. And so being able to take that criticism, not personally, but then use it to make yourself a stronger scientist, that takes a level of tenacity. I think that's a requirement.
I would agree with that. Yeah, absolutely. So you have to be a sucker for punishment in a way, because we do get rejected a lot in all ways, in all sorts of things, and you just have to be willing to believe in what you're doing and to fight for it. There's a famous scientist, and I don't remember her name, but I have a little thing on my board outside the lab, and she basically says something that if you know you're on the right path, you can't let anybody tell you otherwise, and you have to fight for what you're doing, right? So I would second that.
I agree. I actually don't have much to add to that. I agree with all of those. Well, I guess I will add perseverance, being willing to think outside of the box. Sometimes students think that, and Lisa, you mentioned it, they think that maybe they've done something wrong when they're doing these experiments, but sometimes that's your answer, and you made some kind of discovery that someone else didn't see or find. And so you have to be, you have to have perseverance to kind of follow the course, and actually when you get these answers to really pursue it. But then also that would be a personality trait for someone who does want to kind of be more of a independent professor and doing their own research, where you have to constantly ask these questions, and then also persevere when you get an answer that you weren't expecting or thought might be the opposite of what you expected.
And that's, to me, actually is one of the most rewarding parts of doing research, is when you get a result, odds are you may be the first person in human history to see that. And so that's really kind of cool. That's what I find really cool about it, is that you may be the only person in the world who has this knowledge right now. And so that's very exciting. And I think hopefully some of you will share that passion and hopefully be able to make discoveries and find things. And I'll also echo what Leah was saying and Lisa about, finding these alternate observations or having alternate interpretation. That's really important. And that's why it's so important that we have your perspectives, because you approach the world in a different way than I approach the world. And then from Lisa or Leah. And so having different perspectives is really important to drive science forward. If everybody's thinking the same, we're never going to get anywhere, or we're going to end up at a path that we don't want to be. And so it's really important to have a lot of different perspectives, and your perspectives are equally as important as mine. And maybe you'll have some major contributions because you see it differently, and that can really drive us forward. So I would encourage you to be involved as much as you can.
So let's move on to this other question, and it's, what are things that students need to make sure to do to go into a career in research, and what should they be doing now while they're in college?
I'll start. I would say work on your math and science background. Most students will need a major in some type of science field, biology, chemistry, physics. But also, I think when students think science, they forget or don't understand how much math is involved in that also. Mm-hmm. So they need to really kind of beef up their math and science courses. And then also, if there's opportunity for research experience, that's always a plus. And that can be as simple as emailing a professor they know on campus that's doing some type of research they're interested in, and asking to volunteer to just be in the lab and see what's going on. And that could be an open door into getting a research experience.
And that's good for letters of reference too, when you're then applying for post-bac programs. So you need to find ways to have connections like that so that you can have those people write you letters, because that's ultimately very important. And they can make up, so Dr. Rucks can talk more about this because she sits on our graduate recruitment committee, but it's understandable that some students may come in with some deficits in science and math, but oftentimes those can be explained, I think, for some students. And so if you've struggled, maybe because of some personal reasons or family concerns, that can be addressed, and you can speak to that in your own letters, and your references can also hopefully speak to that. Because a strong letter of reference is almost more important than any grades. We're not as concerned about GPA as we are about the character of the person and their desire to learn and be successful.
Yeah, and I would just say, especially right now, there's an opportunity for more virtual sorts of activities. I know that there are several labs that have, say, bioinformatics studies that students can do, which means that they wouldn't be traveling to the lab, but that would still get you involved in that, because it's really the thought process, right, of thinking through. And I would say take advantage of what is local. So say you don't have a lot of research experience, but do you have labs that you can volunteer to help out and to TA in to try to get that sort of experience? I think that if you're innovative in those sorts of ways, that will say a lot about you as a potential applicant, because when we on the grad committee look at applicants, yes, we're looking at GPAs, and if there are certain classes that were struggled in, perhaps there is a personal reason for that, and we look into that. And maybe they didn't have a lot of, at their home institution, lots of opportunities for research. But did they try to maximize what was possible there? Did they volunteer and those sorts of things? So, and I think I said this, I didn't have a lot of that opportunity at my home institution. So I reached out somewhere else to get it. And I was lucky because they were right up the street from each other. So don't feel discouraged if that's not your situation. Try to think creatively about how you can add those experiences to your application, to your experience also, because some of getting the undergraduate experience is putting you in situations where it might look attractive to be in a lab, and suddenly you're in a lab and you're like, maybe this isn't quite what I want to do. It's either going to solidify your goals or you're going to start questioning it again. And that's part of the reason why undergraduate research experiences are important. And so making sure that you're thinking through that, so that can be expressed on your application. And as Scott said, you know, any of these research experiences, if you're working closely with a professor at your local institution and being innovative and helping out with labs, that's going to show through on that letter of recommendation. And so all of that's really important to try to start building your allies in that area.
And let me just add on as well that, as Lisa said, maybe your institution where you are may not have an opportunity for you, but there are lots of summer programs all across the US that you can apply for if you're willing to travel. The National Science Foundation has research for undergraduate, what is it, Research Experience for Undergraduates, REU programs that are at different institutions all across the US. And so there's likely an REU at one of the institutions close to you. And so I would encourage you to try to find those through the National Science Foundation. If you don't have an opportunity at your own institution, there may be something during the summer that you would be able to participate in. And usually those are paid, I believe. So you would be supported. But I think to summarize that, you know, if you are interested in becoming a researcher, then one of the things you should be doing while you are in college is looking for those research experiences. So there are lots of different ways you can go about that, and it's somewhat specific to where you might be and what institution you're at, but trying to show the initiative that you want to be doing it is, I think, important.
Well, let's move on to other questions about what you are all studying. And what are some of the ways that you and other people are doing research in your field, making a difference in the world? You know, why is it important? What type of problem are you trying to solve? Who would like to start?
Okay, I guess I'll start since I heard crickets. So as I mentioned, I work on chlamydia. Chlamydia causes sexually transmitted infections. It's actually the #1 bacterial cause of sexually transmitted infections in the world. So just from that perspective, it's a very important pathogen that causes a lot of morbidity. So it doesn't necessarily kill people, but a lot of young women, if they get an infection, they don't know they're infected, that can cause downstream consequences that could lead to infertility, for instance. So there are a lot of other problems associated with chlamydia that maybe aren't apparent beyond its initial, STD aspect. And so what I study is really the basic microbiology of the organism itself. For instance, most bacteria divide by a process called binary fission, where you have an E. coli that grows and then it splits in the middle and you have 2 new cells. Well, chlamydia doesn't divide that way, and they don't have the machinery, like the genetic machinery, the protein machinery, to do that. So one of the things that we study in my lab is how chlamydia divides, because they don't have all of those things. So, to me, that was already an interesting question. Well, if they don't have that, how do they divide? And then similarly, we also study how chlamydia responds to stress. So when they're inside your body or they're inside a cell, if that cell is stressed out or limited for nutrients, then the chlamydia becomes stressed. And when they become stressed, they're able to basically hibernate inside the cell and resist the stress. And so once the conditions improve, they can come back and start growing again. And that's what makes them a very insidious organism and difficult to treat and eradicate. So we're also interested in my lab in understanding how chlamydia responds to stress, in the hopes that in both of these cases, we can find maybe new therapeutic targets or diagnostic targets that can help us to better treat chlamydial infections.
All right, I guess I will go next since Scott introduced chlamydia. So as I mentioned earlier, I'm a cellular microbiologist, so I study a different aspect of chlamydia pathogenesis. So we mentioned that they are obligate intercellular pathogens. They also grow within a membrane-bound vacuole. So they're sealed off from the rest of the host cell, but they're still manipulating the host cell because they have to get most of their nutrients from the host cell. Part of what makes chlamydial infections difficult to diagnose is that many of the initial infections are asymptomatic. So a woman can be infected for many, many, many months. And that complicates then the treatment of that infection. And so one of the large questions in my lab is, how is it interacting with the host cell, taking what it needs, but then also tamping down all of the normal cellular response, the eukaryotic cellular responses to stress that indicates that something is wrong. And so that for me is a large question. And I think, you know, part of what I've been interested in for a while, but we haven't gotten there yet, is that when women in particular are given antibiotic therapy, it can mess up so much out of the normal vaginal microbiota that can then cause other problems. So wouldn't it be amazing if we could have something that you could treat chlamydia with, and then it would just leave everything else alone? I think that would be amazing. But I also want to stress that I don't go in the lab every day or even every year saying, let's develop a new antimicrobial therapy. I'm really curious about the basic mechanisms happening. And then if we learn something of that can be useful, awesome.
So I actually work on cancer metastasis. So metastasis is when a tumor spreads throughout the body to different places from where it originated. And what we do know as metastasis researchers is that certain tumors will always go to the same secondary organ. There was a researcher towards the end of the 1800s, Stephen Paget, called this the seed and soil hypothesis. And so not all plants will grow in the right or in the same soil and environmental conditions. They require specifics for that. And so what he was basically saying was that there's a certain seed that will always thrive in a certain type of soil. And so, for example, one of the major focuses of my lab is prostate cancer. And prostate cancer and both breast cancer will most frequently spread to the bone more than any other tissue site. And within the bone, there's a whole host of other cells there that can kind of help those cells, those tumor cells, proliferate into a secondary tumor. And so what's always been fascinating to me is how a prostate tumor that's designed to be a prostate cell and a prostate tissue and function in its original site, how it can then move to the bone and act in a completely different way and be able to thrive as a prostate cell in that new soil. And so one of the goals in trying to develop therapies to help prevent cancer metastasis, which ultimately is what kills cancer patients, is to understand how these tumor cells interact with the cells in the new environment. Within the bone is actually a really interesting organ because it's one of the only organs that's always constantly breaking down and repairing itself. And so within the bone, there's a whole host of bone cells that are constantly breaking down bone, building bone. But then there are these immune cells known as neutrophils, which are kind of the front line of defense for our immune system. They will be the first ones to mount a defense against bacteria, tumor cells, and they also will signal to other immune cells to continue on the fight. But for some reason, prostate cancer cells can actually escape the cell killing that neutrophils will induce. And so the focus of my lab is trying to understand how we can disrupt whatever the prostate tumor cells are doing to suppress the neutrophil activity, and maybe we can eventually translate that into clinical practice. Did I answer fully?
Let's, I like this question, but what is the biggest challenge in your field at the moment with what you are studying, what you're working on?
I would say for me, definitely it's not a one size fits all. There's this term that's out there, and especially in the cancer world, about personalized medicine. And there are just so many factors. There's genetics, there's environmental factors, there's socioeconomic factors, and that can relate to when a person knows that they have a tumor. But then there's also therapy that can also change how a tumor functions, either in its original site or secondary site. And so the most difficult thing is that trying to figure out how all of these elements fit into a more generalized answer for what's happening so that we can actually move it on to maybe a therapy is pretty difficult. It's really complicated. And I would say in science, there's kind of a cultural attack against science right now, where people see science equaling fiction, which is absurd. So that kind of trickles down to funding that can go towards biomedical research, and the importance of why we need funding to do our research. So I think just on a larger scale, just understanding that science is critical and the amount of work and complexity to our bodies that there is. So it requires a lot of work and time for us to try to figure out what's going on.
Dr. Ouellette or Dr. Rucks? Major challenges?
Yeah, I agree. I think there are maybe two big challenges I see right now, and it kind of relates to some of the other questions, so maybe we'll talk about it more. One is having people. So this is where you come in very important, because we need more people who are interested in doing research, and who are passionate about learning and want to contribute in that way. So having the personnel, so the human resource aspect, I think is very important, and we need more people who want to do science. The second is that with some of the new technologies that we have, we're generating reams of data, like silly amounts of data. And now the big problem is trying to interpret it. You have so much information, and it's just like, how do you process it all? And maybe we can talk more about that in a little bit, but do you have anything to add, Lisa?
No, I'm actually, I don't know. I'm just so excited about what's happening in the lab right now that it's hard for me to really focus on the challenges so much. But we can talk about all the exciting stuff in the next couple of questions. But I think that this is also very important, like getting students in the lab that are really interested in doing science and wanting to learn is important. And then also, I think in general, we need to become better communicating, like what Leah said, with the general public about the importance of what we're doing, even at a basic science level. So that there is more support, not just for the science and believing in the science, because we're seeing that's a huge problem right now, but just funding the science so we can keep doing what we're doing.
So let's talk about the changes in your industry in the last 5 years and what you predict is going to happen in the next 5 to 10 years. Dr. Ouellette, you were alluding to some of this, that there's specific technologies that are happening. So can we talk a little bit more about these two questions that were asked by students?
Sure. Yeah. So these are all great questions that you guys are asking. So I applaud you for them. You've given them some careful thought, but certainly, as I was indicating, we have all of these machines now that can help generate a lot of data. So whether it's genomic data, so we can sequence people's genomes and have all of the information about different people, and even specific cells. So like Leah might be interested in, what's the tumor DNA look like in this particular cell type. And so we have all this genomic DNA, and then likewise we can generate lots of transcriptional data, so the mRNA that eventually gets translated to proteins. And so we have lots of transcriptional data that can be generated, lots of protein data that can be generated. And as I mentioned, the real problem over the past 5 years has been, how do we interpret it? And this can speak then to the next 5 or 10 years, is that I think what we need to be able to do is, because these technologies are going to continue to advance. And so we're going to continue to be able to generate lots and lots of data. And so what we really need then are, and maybe this ties back to one of the earlier questions, is how do we interpret it? And I think if you are interested in computers at all, and you have a skill in coding, then having that ability to bridge from computers and doing bioinformatics to the biology at the bench is really going to be critical for us to help understand all of those data. So bioinformatics, I think, will be a big field that's very important. So if you have an interest in biology, but you're actually more interested in computer science or mathematics, I would encourage you to consider developing those computer skills to apply them to biology, because that's going to be very helpful for us. Unfortunately, I don't have those skills, and so I look at all these data going, what now? I don't know what to do with it. And so that's a big challenge. And I think it's also affecting a little bit how we do science. So, broadly speaking, there are two types of ways to do science. One is you just generate the data, and then you generate more data, and you just continue that without really thinking about it or what it means. And so I kind of call that factory science. That's not so appealing to me. What we prefer to do, or what I prefer to do, is to have a hypothesis, right? So we have a specific question we're trying to answer, and then we go out and find the ways that we can test it. And that lets us then have a specific goal in mind when we're interpreting data too, because when you have all these data that are being generated and you don't know how to interpret it, then it really makes things very muddy and unclear. So I think it's changing the way we're doing science, and not necessarily for the better. So if you're interested in going to science, I'd always encourage that hypothesis and question asking aspect of the process. And don't set that aside. That's really what would make you a better scientist overall. Anybody else have?
I think what we're going to see improvements on are models of infection. So with chlamydia, the mouse models, there are several mouse models out there, but they don't really recapitulate the type of infection that happens in humans. In mice, it tends to be really aggressive, which is not what happens. And mice have also different signaling pathways that get activated that human cells simply don't have, which makes those data difficult to relate to what's happening in humans. But we have several different cell culture models that are emerging that are kind of layering different types of cells on top of each other, that almost recapitulates what you might see in a microenvironment, in an actual infection microenvironment, whether it's the endocervix or the uterus or even the fallopian tubes or oviducts. So that's really exciting. I do a lot of imaging, although I don't want you to have the impression that my entire day in the lab is at a microscope, because it's not. But we've had a lot of really cool advances in live cell imaging. And so in the upcoming months in my lab, I'm so excited about to jump out of my skin about it. We're going to be able to look at what eukaryotic proteins are being trafficked to the inclusion, and see if they stay there or if they actually pop off and traffic back into the eukaryotic cell, which would be super exciting. But our limitations there are, whenever you have a fluorescent protein, it only lights up for so long. And so that's your finite time of experimentation. And so I think in the next 5 to 10 years, we're probably going to see improvements in that technology for longer lasting fluorescent proteins. And then the ability to go into single cells and really do, so you're doing all of your imaging experiments in this one cell, the ability to then take that cell and analyze it genetically or by proteomics or something of that sort. Lipidomics even, lipids are really important to my research. I think that's where we're going to be heading in the next decade. It's really exciting. It's an exciting time to be a chlamydia biologist. What can I say?
But you're not biased though, right?
Not biased. Mm-hmm.
I would say for cancer biology, I think we've transitioned in the past years to looking more at the microenvironment. And so a lot of our early studies were focused more on the genetics and the protein profile of these tumor cells and what could contribute to them becoming a transformed cancerous cell from a normal epithelial cell. But what a lot of research is noticing is that, similar to what I'm looking at, is that these cells are kind of in an ecosystem with other different cell types. And all of those cell types work together to promote tumor growth. And so a lot of really new technologies are coming out to look at these full cell populations and to get genetic information from all the cell populations within a tumor or from a person's blood sample. So for example, this technology single cell RNA sequencing. And a lot of cancer biologists and also just all researchers are starting to use more of this technology. But what it allows for us to do is to look at the components of all the different cells in that population to see how they are different from what we thought. And so even when we just think about immunology, there's a lot of work going towards how we can utilize the just immune system to attack the tumor cells, which they should be doing. But we're also starting to see that different immune cells, they act in different ways depending on what tissue they're in or what tumor they're encountering. And so with technologies like single cell RNA sequencing, we can get that information and say, well, how does this tumor look in the bone compared to what it looked like in breast tissue? And what are all the cells involved in that? And so I mentioned personalized medicine, and I think that our field is actually moving to more of that with some of these technologies. Now, with what Dr. Ouellette mentioned, it also is a lot more information. So then we have to try to figure out what it all means and in the big picture of things, what it means functionally for a cell. But it is pretty exciting because we're starting to see a lot of things that we thought based off of previous research, which got us here, but we're starting to see that these cells are really dynamic and they seem to just want to survive. And they will interact with other cells, however, in a way that will promote survival, however that may be. And also therapies can also do that. So it's pretty interesting how it seems like the cancer field seems to be overturning a lot of the dogmas that we originally thought about cancer biology to really look at it more on a personalized level and see how these different interactions that we see depending on the patient can inform us of cancer as a whole.
I just want to add one thing to that discussion. So both Lisa and Leah mentioned microenvironments, and I agree with them that this is an exciting new area of trying to understand in a more holistic way the whole area that you're interested in. And this applies to more than just biomedical research. So we're all, I guess, technically biomedical researchers. But if you're interested in soil or you're interested in water chemistry, there's an opportunity there as well to better understand what role the microbiome, for instance, plays. So we have a side project in my lab where we're very interested, as an example, in looking at insect microbiomes and how those can be impacted by a specific type of bacteria called Wolbachia that can adjust sort of the sex ratios of the insect. And then that can have downstream consequences potentially in terms of what food supplies the fish might have in the rivers, or birds once those insects come out of the water and look to mate. So thinking mayflies or stoneflies. So having that more holistic understanding of the topic of study, I think, is going to become more and more important as we go forward, because we have the ability now to analyze lots of things. Again, the drawback is that it means we have lots of data that then need to be interpreted and analyzed. But if you're not interested in biomedical research, that's fine. I mean, that's great. You have your own interests, and I would encourage you to think more holistically about those areas, because there's microbiomes in soil, for instance, that can impact the soil health or the water quality. So there are lots of these microenvironment types of questions that apply beyond just what we do in our labs.
So we have 2 questions left. And now I'd like to cover this idea of misconceptions that people have about research career. And how do you think that we can combat these misconceptions and communicate them a little bit more effectively to people?
Maybe we should have asked what their misconceptions were. Yeah, I'm not sure how to answer that. Does somebody want to take a shot?
I'll take a shot at it. I do feel like, and I always feel this way whenever I see science being done in a TV show or in a movie, you see one of 2 things. You either see this really glamorized lab where there's so much really expensive equipment, even though like I know it's expensive, but the general public doesn't. I'm like, that's not realistic. you know, and then you have the scientists that, especially if it's a female, then that person will have a certain type of hairstyle and be really, really fashionable. And I think there's this kind of equating of occupation with how you look often. And then you have what most people think of the real world, where they feel like scientists always wear glasses and don't really care about their outer appearance, and they're stuck in the lab all hours. And I feel like that's one of the myths, is how we look as individuals, when it has nothing to do with that at all. And I actually am guilty of that. When I went into my first lab, one of the reasons I liked it so much, because I was like, wow, these people are normal. And that was one of the things. We would go on group lunches, and during the summer, our professor, he would take us canoeing and kayaking, because he was a summer lifeguard when he was younger. And so he would always encourage these group outings where we would do a lot of water things. But these were just normal people who were really interested in science, and they had regular lives. So I think that's a myth, is that scientists are just these kind of nerds that are bunkered down. Now, mind you, I do consider myself a nerd. I'm interested in science and, you know, just really complicated things, but that shouldn't be equated with being a negative. Whereas this culture of whether it's cool or not, that should have nothing to do with people's conception of what scientists are doing. And then also, science is not political. So science is just fact. It is what it is. And I do think that's kind of being lost right now. You guys might not hear as much about it as some of the adults are, but it does seem to be more associated with opinion and what people think is real versus what is real. And so I think that's also a big misconception that's out there, that science is up to the believer and whoever is thinking about it. But really, science is one of the few things that we have that is fact.
Yeah, I'm going to piggyback on what Leah was saying about some of the depictions of science, like in popular shows. I'm going to pick on CSI right now, because every time they pop something in a centrifuge, and an hour later they have an answer. That's not how science works at all. We do use centrifuges, but usually as a tool to separate bits from each other. But your answers in an experiment, it can take weeks, sometimes a long period of time. And so there's an element of patience that's required when you're a scientist, that I think if you're married to that popular notion of what science is, the data are the data, and they will take their time. They don't care about your grand plans for when to be done. So I think that that's one of the misconceptions. Yeah.
So let me add about this combating part, because this can maybe lead to another career option. So I think the way we combat these is through communication, right? And there is a career where you can be a science communicator. Some people who are very passionate about outreach, but maybe don't want to be in the lab anymore, if they have a PhD, they can be effectively a science communicator, or somebody who works with their elders or their Congress members so that they help those people understand science more. And I think that's another important thing that we need. So if you are somebody who is passionate about science, but maybe along the way you've decided, well, you know, I don't know that I want to work in the lab anymore, then that could be a career option. So I always like to tell people that if you have a PhD, it only opens doors for you. It doesn't hurt to be more educated or to have more credentials. So if you have a PhD, there are lots of things you can do with it. It doesn't mean you're automatically tied into being a researcher. You could go work in law, for instance, as a patent law lawyer. And that's a very lucrative industry, and they would even pay for you to go to law school, is what I've understood about it. But there is the ability to be a science communicator, and I think that's one way that we can help combat some of these misconceptions. And I honestly am not aware of what sorts of misconceptions people may have, perhaps because I'm too focused on what I'm doing myself, and I'm not so hip with the cool kids.
And finally, the last question is, what is one piece of practical advice that each one of you could give to students that are wanting to start out a career in bench research and maybe set off in a similar direction as one of you? So what type of advice would you give to college students?
Don't be afraid to knock on that closed door that you see in front of you. Don't be afraid. Just go for it.
I agree. I mean, you have to be willing to take a shot and take a chance. And that can be scary, you know, but we all had to do it at some point. And as I said, I'm the first scientist in my family. So, you know, I had to do that. And probably Lisa's told you how she did that, and Leah's done that. So everybody has to do it at some point. And on the flip side, though, don't be discouraged. Don't take no for an answer necessarily. So maybe that door didn't open, but there is a door that will open. And so keep knocking. If you're passionate about it, you can't give up.
Yeah, I agree with that. And to add to that, don't be afraid to pursue science when your friends and family aren't interested in it. As you mentioned, Scott, I'm the only scientist in my family, likely the same for you, Lisa, and my family thinks I'm weird. They don't understand what I do. And if I ever listen to them as far as what they thought I should do versus what my passion said and what I wanted to do, I probably wouldn't be a scientist. So don't be afraid to think outside of the box and pursue what you're interested in. If you're interested in science, go after it and pursue some of these opportunities that we've talked about.
And I would just like to say that, you know, Leah mentioned that one of the things that she was interested in was that, you know, prostate cancer disproportionately affects African-American men. And, you know, there's a lot of science and diseases that maybe disproportionately affect Native Americans. We know that diabetes, sexually transmitted infections, for instance, are also oftentimes elevated in reservation communities. So, you know, if you're not interested in anything that we've done or talked about, that's totally fine. There are many ways you can find research that will help your communities. And I would encourage you to look into those and to find them and find your passion.
And it is clear that the three of you are really passionate about what you do and are excited about everything you're working on. And I do appreciate the time that you took today to share some of that passion, some of that advice, and clarify some of these questions that students had for all of you. So for now, we will be ending the call and stopping the recording. Thank you very much.
Thank you all.
Thank you.
Thanks.